Hi, everyone. I'm Mark Johnson, a volunteer with the Canadian Celiac Association. Welcome to the latest edition of the In Conversation With series. Today, I'm going to be talking to Dr. Heather Galipeau, who is the latest winner of a research grant from the Canadian Celiac Association. Welcome, Heather.
Hello.
So for everyone's background, this grant comes from the CCA's J. Alexander Campbell Research Fund. So this is a grant of up to $25,000 to support scientific research in Canada relating to celiac disease or dermatitis herpetiformis or non-celiac gluten sensitivity. Some researchers also look at areas like the social and economic implications of delayed celiac diagnosis or living with these conditions. So now, having set the stage, I'm thrilled to be speaking with Dr. Galipeau today. She got her PhD from McMaster University in 2015, and she currently holds a position as Research Associate and Assistant Director of the Axonic Notobiotic Unit at McMaster. Dr. Galipeau's research focuses on mouse modeling and other models that are looking at the immune reactions in chronic inflammatory diseases, like celiac disease. And specifically, she's researching how bacteria can affect the triggering of celiac disease, looking at what can cause someone to become celiac. Her long-term objective is to develop therapies to prevent or treat chronic inflammatory and autoimmune conditions like celiac, something we can all get behind. So Dr. Galipeau, let's dive right in. Your research title is A Novel Model Using Organoid Technology to Study Epithelial Mechanisms in Celiac Disease. In plain language, could you tell me what that is?
Yeah, so that's a bit of a mouthful, but basically we're using, I guess, what we can call mini guts. So these are basically intestines that are grown in petri dishes to study how the cells that line our gut are really involved in celiac disease. So that's kind of a one line of the overall aim of the project.
So growing guts in petri dishes. It sounds a little bit like science fiction. So
Yeah.
why exactly are you doing that?
Yeah, so it's another way to model the disease. It's more simplistic than an animal model, but more complex than conventional approaches that use cells in a petri dish. So past approaches have used cells that don't really mimic the full intestine. And when we grow these mini guts, basically we can isolate cells from even a human or a mouse that can express the genetic risk genes for celiac disease. And then we grow them in a specialized way that they basically recapitulate many of the features of the gut. So they have all the different cells that make up our small intestine. And what's really important and really cool is that when we use these engineering approaches to more mimic the environment in the gut, we can get the villi that are really characteristic of the small intestine and really important in celiac disease pathology.
So is this kind of one step away from growing organs, or is this something completely different?
It pretty much, so what we also call them organoids. So they're mini organs that are grown in petri dishes, and you could do it with other organs other than intestines. So you're spot on with that.
Okay, analogy. And I know the bacteria in the gut is like incredibly complex. You know, I think we're talking billions of cells, and that, like, how can you reproduce something so complex with accuracy?
So the organoids, the mini guts, they recapitulate the human side, not necessarily bacterial side, but you can introduce, so then when we use another technique called the gut on a chip, you then take these 3D structures and put them back onto a chip basically, and you can introduce different bacteria into the system. So again, it's a more simplistic way that we can study really specific interactions between specific microbes and our epithelial cells.
All right, and epithelial cells are what exactly?
So these are the cells that line our gastrointestinal tract. There's different types of cells, but they're what form that barrier between, I guess, the outside world and inside of our body.
Okay, I know that it's the little hairs in the small intestine that get damaged and destroyed when someone with celiac disease eats gluten. But you're not looking at those. You're looking at a separate layer of cells that coats the small intestine. Why is that?
Yeah, so it's actually the cells. The cells will actually form these projections, the villi of the small intestine. So that villi is made up of all these different cells. And when someone who has celiac disease, that villi will become flattened. And that means that they're going to have problems with nutrient absorption.
Yeah. Okay, so it's really one and the same. These are extensions
Yep.
of these cells that, okay,
Yep.
so that's interesting. Okay, let's thanks for indulging me on that. Let's to back up a little bit. I know you've talked about one of the reasonings for your research is because the gluten-free diet is difficult to follow. Can you explain what you mean by that?
Yeah, so I mean, there are more and more options for gluten-free options out there, but when you have celiac disease, you have to be, I mean, completely gluten-free. And many times there's cross-contamination with foods, and really, really small amounts of gluten can cause symptoms in patients, and also mucosal inflammation, so persistent mucosal inflammation. And that's not good, obviously. This can lead to nutritional deficiencies, bone deficiencies. And I guess another example is when you're eating out, you don't really know if there's these really small amounts of cross-contamination. And again, frequent cross-contamination is one of the main reasons that people continue to experience symptoms and inflammation in their gut. And all this really causes added stress to people with celiac disease. So because it's so hard to follow, we really want to be able to develop additional treatments to the gluten-free diet to help people so they don't have these persistent symptoms, so we don't have this persistent mucosal inflammation in the gut.
Okay, so this will help everyone with celiac disease hopefully down the road, but it's really the motivation is the folks that are diagnosed and are eating gluten-free, but for some reason they're still feeling terribly and suspect that they're getting gluten, but they can't figure out where and suffering ongoing symptoms.
Yeah, so I think there are statistics out there that up to 60% of people will still have symptoms even though they're following the gluten-free diet, and it's just as many that will have persistent inflammation even after 5 years of following the gluten-free diet. So that's a long time to be following the gluten-free diet and still having inflammation ongoing in your gut. And so that's something that we want to help people. So.
And we know celiacs often have other ailments. Unfortunately, the disease often isn't alone. I know you're looking at intestinal inflammatory diseases. Are you looking at other ones besides celiac disease? Which ones would those be?
Yeah, so the other main focus in the lab is IBD, so Crohn's disease and ulcerative colitis. And yeah, so we're also looking at how microbes are involved in triggering IBD. So that's the other, I guess, main focus of the lab.
So this the research with the miniguts might help contribute to therapies for a variety of intestinal diseases.
Oh yeah, for sure. Yeah.
Okay, sounds great. And I know in your lab, we've heard at conferences of the Celiac Association before about the research going on there with the mouse models. Can you, like for the non-scientific among us, can you explain why use mice for the models?
Yeah, so that's a good question. I mean, for the most part, they're easy to work with. We can also genetically modify them so that they express the human celiac risk genes. So we use mice that express the human DQ8 and DQ2. So those are the genes that predispose you to get celiac disease. And then also we can manipulate, for example, the bugs that are present in the mouse gut. So this allows us to really study specific mechanisms involved in celiac disease. So they can be humanized from a genetic standpoint. So they express the human celiac risk genes, and then humanized from a microbe standpoint, where we can put in bugs, for example, that come from a celiac disease patient.
And you said that the new mini guts in the Petri dishes are going to give you more accurate results. Was that right?
So they'll give us more, I don't know if I'd say more accurate, but they're a better way to study celiac disease than previous approaches using cells grown in Petri dishes that don't necessarily come from the small intestine. They don't form the villi structures like we have in the small intestine. So they're not necessarily representative of a human small intestine. And that's where these mini guts are more advantageous than past approaches. But really they complement the mouse work. So you can study mechanisms in the mouse, but then really tease apart what exactly is going on when you go to this culture approach.
I'm risking a very sciencey answer here, I know, but are you able to talk about, just out of curiosity, what are some of the chemicals, like the building blocks of these mini guts, or is that protected information?
Well, I won't go into specifics, but basically what you do is you isolate stem cells from your organ of interest. So we'll isolate stem cells from the small intestine of our genetically susceptible mouse, so a mouse that expresses the DQ8 or DQ2 gene. And then we basically just expose them to certain growth factors. And that's going to cause them to grow in a specific way that will form. Basically, it's this 3D structure that recapitulates many aspects of the intestine. I won't go into specifics about the growth factors, but
oh, it's fascinating. I know there's a lot of research going on with adult stem cell research. Are you using any of that, or is it all from the mice because they've been conditioned to be celiac?
Yeah, so we use the mouse cells. You can grow these mini guts or organoids using cells that come from human biopsies. So for example, if someone's going for an endoscopy, you can isolate cells from that. But that becomes much more challenging because you're dependent on getting these samples from a clinic. You can't you can't manipulate the tissue beforehand, whereas in a mouse, we could, for example, expose the mouse to gluten before and then take the cells. And that might be different from a mouse that wasn't exposed to gluten, whereas you can't necessarily do that with the human tissues.
Okay, I think you've answered my next question, that celiacs, we're often watching for opportunities to help with research. How can we contribute? Can we sign up? But that isn't really appropriate for the particular research you're doing.
No, but there are, I mean, if you're attending the celiac clinic at McMaster and you're asked to participate in a study, they're always looking for volunteers for clinical studies. McMaster has the adult celiac clinic, and they have lots of ongoing research there.
Right. Yeah, I know a lot of our members have taken advantage of that, a beacon of light at McMaster. So I know to get back to the cells that you're looking at, the epithelial cells, I know you're looking at those as one of several factors that could impact the immune response that happens in celiac people. What are some other factors that you're looking at?
Yeah, so the major other focus is how microbes can contribute to celiac disease. And so that's really been the focus to this date of my work in the lab. So not necessarily what bacteria are there, but how these bacteria are functioning. So for example, certain bacteria can degrade the gluten peptides that we eat, and they can degrade them in a way that make them either more inflammatory or less inflammatory. Then we have other bacteria that can degrade other components of our diet, like the amino acid tryptophan. And these products are actually anti-inflammatory. So you can see that you can, depending on what microbes might be present, but I guess importantly, what these microbes are doing, you can either have a situation that is more inflammatory or a situation that's less inflammatory. So we're really interested in studying kind of how these functions can lead to the development of inflammatory responses towards gluten.
Okay, so I mean, everyone listening is always wants to know, like, what's in it for me? How's this going to help us? And we've covered a lot of the science, but so to make a big leap to, 5-10 years down the road, after your research is wrapped up, when you in terms of looking at how various bacteria can make gluten particles or whatever more inflammatory or less, is the goal here to identify what can make gluten less inflammatory that might let someone with celiac disease eat gluten safely down the road?
So,
or is that too much of a stretch?
I mean, ultimately that would be amazing. But that would be a lot of gluten for your bacteria to break down. But it might be used in more so the case of somebody who's experiencing these persistent symptoms or inflammation. And so they might be getting really small amounts of gluten. And perhaps if there was a way to break down that gluten into these less inflammatory peptides, that might be a way to help them feel better.
I think that's a message we've heard several times, that these treatments are not meant to let you go pig out at Tim Hortons, but it's more about protecting you from accidental gluten exposure at a restaurant or somewhere.
Exactly, yeah.
Mm-hmm. So what do you hope comes out of this specific research that's being funded with this grant?
Yeah, so I mean, ultimately, we hope that it will shed some light on the initial triggers of celiac disease. So how somebody with the celiac risk genes and who eats gluten, how that turns into this inflammatory response that leads to this villus atrophy. And why is it that somebody with the same risk genes and eats gluten doesn't? So why is there this difference? And ultimately, this is going to help us come up with preventative strategies and hopefully new targets that we can target therapeutically.
There's various therapies going, like research into therapies going on for celiac disease. I'm sure you're well aware of them. So what kind of what gap does your research fill in that world?
Yeah, so that's a good question. A lot of the research on therapies coming up now, I guess, aren't really targeting the epithelium, or they're targeting somebody who might already have established disease. And this project is really looking at the initiation of disease. So what causes that initial response to gluten? So this will really help us come up with maybe some preventative strategies, as opposed to, for example, you might be aware of the vaccine for celiac disease, where they're trying to reverse somebody who already has celiac disease, make them non-responsive. So I think that's where this might be a little bit different.
Okay, and for our listeners, don't get too excited about the vaccine. We're a long ways away from being able to line up for that vaccination, unfortunately. But it's exciting. So I know, this sounds like such a massive undertaking. I know your lab's been working on various aspects of this research for years, and the mouse model. How large is your team?
To think. So we've got a couple postdocs, quite a few PhD students and master's students, research assistant, lab manager, and then our technicians who work in the xenic autobiotic unit as well. So we're a good sized team.
And what's the time frame? Like you receive the grant, I know it helps to support your research, and I assume that you'll be looking to publish findings down the road. Do you have a rough idea? And I know you can't guarantee any timelines, but do you know how long it might take before we're at that stage where you're able to publish your findings?
It's really hard to say. Hopefully within the next year or so, we'll be able to have something out. Hopefully with COVID restrictions, it doesn't slow us down too much, but hopefully soon you'll hear something from us.
How has your team adapted in the COVID area? Era? I'm sure this is difficult research to do remotely.
Yeah, so I mean, we have been fortunate in that we've been able to keep research going just at a slightly reduced capacity. So, we have to limit the number of people in the lab at one time. We have a cap of the number of people who can be there. So we work in shifts from the work that we can do at home, reading papers, analyzing data. We do that from home, and we have our weekly Zoom lab meetings now. So we've adapted, but it will be nice when we can go back full force.
Yeah, it is at McMaster. So like you're working in the research, of course, but do you have members of your team that are doing this as part of coursework or towards their own doctorates? How does that work?
You mean working in the lab as part of like a master's or PhD degree? Yeah, so we have a number of students involved in celiac projects who are doing both a master's and PhD. And we usually have a couple of undergraduate students as well who are doing like a project for their 4th year of undergrad, which is actually how I started in Elena's lab. So.
Oh yeah, so that's Dr. Verdu for our listeners, Dr.
Yes.
Elena Verdu, who's a familiar face to the CCA. I know you, Dr. Galipeau, have been a past recipient of funding from the CCA's J.A. Campbell Research Fund too. Can you remind us a bit about what that research was?
Yeah, so I was very fortunate to get the Young Investigator Award a number of years ago, and it really helped me in getting my PhD. So that award was used to really help in the development of some of these animal models that we continue to use in the lab today. So specifically, it was the mice that express the human DQ8 celiac genetic susceptibility gene. And we were looking at how the presence or absence of certain microbes could influence how these mice respond to gluten. So that was, I think, the focus of that project.
It's exciting to see how my previous research folds into the next step and the next step. And
yeah, so we're still using the same model today. We've just adapted it, and yeah.
Okay, well, hopefully we'll be able to speak with you again once you're wrapping up your research to hear about the findings.
Yeah, it would be amazing to be able to share our results with you when we get them.
I'll look forward to that. And this has been fascinating. Thank you very much, Dr. Galipeau, for joining me today, and congratulations on being selected for this research grant from the CCA.
Oh, no, thank you so much for asking me to be here, and especially for this award. It means a lot to me, and I'm really excited to do this research and share it with you.
All right. So is there anything else you wanted to add, or we about covered it?
No, I think that's good.
Thank you. I did want to bring it all together for the listeners with some background on the J. Alexander Campbell Research Fund of the Canadian Celiac Association. So it was named after the late J. Alexander Campbell, who he worked for the government of Canada actually for a time in agriculture and in health. He was chair of the predecessor to the Canadian Celiac Association's Professional Advisory Council, which we have today. Dr. Campbell really helped pave the way to proper gluten labeling on food, and he helped develop the CCA's Pocket Dictionary, was involved in their earlier days of the CCA. So he was really a huge asset to the celiac community in Canada and laying the groundwork to where we are now. So the research fund named after Dr. Campbell is used to continue the work that he started. like investigating and reporting on all the various aspects of celiac disease and the gluten-free diet, and to encourage others in the field who share this interest. So each year, the CCA's board of directors awards grants, like the one that Dr. Galipeau is receiving, based on advice from the Professional Advisory Council. So they review applications for funding that come in. And since 2000, the CCA has given out almost $400,000 in research grants through this fund to support Canadian research of importance to us. So I'm really glad that we're able to talk about the latest research being supported by the CCA. And thanks again, Dr. Galipeau, for taking the time to speak with me and for all the work that you've done and continue to do for our community.
Well, thank you for having me. And again, for all the support of the CCA, we really appreciate it.
And we appreciate you. Thanks, and bye, everyone. Thanks for joining us.